Hi. Today what we're going to talk about is we're going to talk about two topics. One, restriction enzymes, and the other, plasmid cloning. Now, they're tied together because the way we're going to talk about plasmid cloning is we're going to need restriction enzymes. Now, these are two different topics. We're going to unpackage these terms as we go. So essentially, the end goal of doing plasmid cloning, what we're trying to do is we're trying to incorporate a piece of DNA into a plasmid, which is a circular piece of DNA, that plasmid is then going to get put into some bacteria, and then we can grow up that bacteria and then therefore grow up more of that particular fragment of DNA. And so it's a way of isolating out individual small pieces of DNA that we can use to study or for various applications. And then we can just, if we want more of our material, more of that particular sequence, we can just grow up more bacteria, extract out the plasmid, and isolate the piece of DNA. Now, in order to get that piece of DNA into the plasmid that we're as from this particular method, is we're going to need to use what's called a restriction enzyme. Now, restriction enzymes are a class of enzymes called endonucleases. So what an endonuclease is, any nuclease is an enzyme that degrades nucleic acids. An endonuclease means that it degrades nucleic acids from within, and rather than the ends of the nucleic acid, and so it doesn't start from the sides and work its way in. It can cut from within the sequence. Now, the interesting thing or the unique thing about restriction enzymes is that they recognize a particular sequence of DNA. And so these are found in different types of bacteria. They're thought to have evolved as a defense mechanism, probably against invading other DNAs, probably maybe from bacteriophages or viruses of some sort. And they recognize a specific sequence of DNA and will cut the DNA at that particular sequence. Now, that sequence recognition is like a lock in a keyhole. And so a particular sequence of DNA is a three-dimensional molecule, and one sequence of DNA is going to have a different shape than another sequence of DNA. And so restriction enzymes have evolved to bind onto a particular sequence of DNA, so they have a particular shape that they can fit onto, like a lock and a key. And so if you change that sequence, you lose that interaction. The key no longer fits into the lock. So the first worksheet we're going to look at is just looking at restriction enzymes and then evaluating and visualizing what happens when we cut up a piece of DNA with restriction enzymes. To do that, what we're going to use is what's called gel electrophoresis. So gel electrophoresis is a way for us to visualize our DNA, and we're going to do that by separating the DNA based on size, and then we can visualize them, and we can make sure that we have the DNA that we expect to have, that it's of the size that we would expect, and then we can and that way we know it's there and we're working with the right thing. Now, gel electrophoresis, what we're going to do gel electrophoresis, the gel is a gel matrix. It's like a gelatin that we're going to stick this comb into that makes these dents into the gel. Okay, so that's what these squares are at the top. They're what we would call the wells. That's where we're going to put our sample, and then what we do is we're going to put this gel matrix into a buffer, and that buffer is going to it's a liquid that will allow electrical current to move through it. So we're going to run an electrical current through this gel, and then with a positive electrode at one end, a positive electrode at one end, a negative electrode at the other end, and then the DNA, being negatively charged, is going to be attracted to the positive pole. Okay, so it's going to get pulled through the gel matrix towards the positive pole, and this is like a sieve of sorts in which it's a matrix that the pieces of DNA need to find their way through. Smaller pieces are going to move faster than larger pieces. So as we run the gel for a length of time, the smaller pieces are going to end up at the bottom. The larger pieces are going to end up at the top. Now, to determine what size the fragments that we're evaluating are, we always need to include what's called a size standard or a ladder. Now, the size standard or the ladder is used because the rate at which that you move that the DNA moves through the gel is going to depend on the size of the piece of DNA as well as the voltage that you use and the density of the gel. And so since different factors are going to be involved, we need to have some comparison to something we know. And so what we have here on the side is what we call is an example of a size standard. And so in this case, this size standard is every 2,000 base pairs. KB stands for kilobases, so 2 KB is 2,000 base pairs, 4 KB is 4,000 base pairs. And what we're going to do is we have different situations in which we're going to cut this piece of DNA with these particular EcoR1 sites. So what are EcoR1 sites? These are restriction enzyme sites. So restriction enzyme sites have a particular sequence. The sequence that that EcoR1 recognizes is actually GAATTC, and this is a palindromic sequence, meaning that if I have 5' to 3', both of the 5' to 3' direction sides of the DNA, both sides of the DNA, when read in the 5' to 3' direction, are the same sequence. So if I go 5' over on this side, it's going to be GAATTC 3' as well. Okay. Now, EcoR1, it's going to recognize these sites, and it's actually going to cut at that location and at that location. And this is what forms what we call compatible staggered or sticky ends of our DNA. And so what the DNA would look like after we cut it, and then we'd have, and so the AATT here is complementary to the TTAA, and so it's possible for these two to rehybridize together, and that's what we're going to take advantage of when we talk about plasmid cloning. But for now, all that we're going to do is that these are just two sites that we can cut this DNA. Now we're going to cut the DNA, and when you're cutting DNA with a restriction enzyme, what you want to keep in mind is, are you cutting a linear piece of DNA or are you cutting a circular piece of DNA? And so in this example, we're cutting a linear piece of DNA. So cutting a linear piece of DNA is like cutting a string. Okay, you got a piece of string, and this is a string, and we got 2 KB, 4 KB, 5 KB, so 9, 11 KB in total length. So if you think about cutting a string that's, say, 11 inches or 11 centimeters, whichever your unit of choice may be, say 11 inches in length, and you cut after 2 inches, and you cut after another 4 inches, you'd be left with a 2-inch fragment, a piece of string, a 4-inch piece of string, and a 5-inch piece of string. Okay. In this case, we're talking about bases of DNA, thousands of bases of DNA, so KB. So we have a 2 KB, 2 kilobase, 4 kilobase, and 5 kilobase fragment when we cut this piece of DNA with EcoR1. Now, when we're using these, what this activity does is considers what would be the banding pattern that would result in the gel from cutting up this piece of DNA in different scenarios. Now, before we do that, the other thing to consider, which we'll look at in more detail in the next activity, is what happens when we cut a circular piece of DNA. And so if you have a circular piece of DNA that has a single restriction enzyme site, what you're going to do is just linearize and just cut open that circle like cutting a rubber band. You're going to make one cut. That just makes you have a linear piece of rubber. Okay, and so we'll look at that in a minute because that's what we'll do when we talk about plasmid cloning. But for now, let's see if we solve what look what this gel would look like. So here we have these restriction enzyme sites, and so for this first one, we're cutting this piece of DNA that has both of these restriction enzyme sites. And we're going to stain it with ethidium bromide. That just allows us to illuminate the DNA with ultraviolet light. And then what would the resulting gel look like? And so to do that, what we're going to have is a 2 KB, 4 KB, and 5 KB band on our gel. And so that would simply look like this. We'd have a band at 2 KB. We'd have a band at 4 KB, and we'd have a band at 5 KB. Okay. Now the next example, or the next problem shown here, is now if a mutation alters EcoR1 site 1 and it's no longer functional, what would be the banding pattern? How would it differ? And so what that means is EcoR1, the first restriction enzyme site that we have here, doesn't work. Okay. So what that means is we're not going to cut there. So I'm going to have this 2,000 base pairs and this 4,000 base pairs. So I'm going to have 6,000 base pairs altogether. And then I'm going to have 5,000 base pairs. Now, again, we're going to do the same thing that we did for the other one. We're going to use our size standard. And so here we're going to need this 4,000 and 2,000 is 6,000. So there's our 6,000. So lining up with this lane, we have a band at 6,000. And then we also have that 5,000 base pair piece. So we're also going to have a fragment right there. Okay. Now, for part C, so this would be this is A, B, C, D, and E. And so for part C, if a mutation alters sites 1 and 2, so they no longer function, what will the banding pattern be? Well, in that case, what we're going to have happen is we have neither of these sites are going to work. And if that happens, then we don't cut the DNA at all. And so instead, we're just going to have one piece of DNA that is of the original length, in which case will be about 11 kb, which will be about there. Okay. And now for this next one, if 1,000 base pairs of DNA were inserted between the two restriction enzyme sites, how would the banding pattern in the gel look? Okay, so what that means is we have the 2,000 base pairs, and then we insert another 1,000 base pairs here. So we have 4,000 base pairs plus another 1,000, so that's 5,000, and then this one's 5,000. And so the way this would look on the gel is when we cut up this piece of DNA, we're going to have lots of copies of it in there, but every one of them is going to be cut in the same spot. We're going to cut here and we're going to cut there, but this middle piece is the same size as that. The last piece, that 5,000 base pair piece. And so we're going to have twice as much material at the 5,000 base pair mark than the 2,000 base pair mark. And so we would still have a band at 2,000 base pairs. And then we'd have a band at the 5,000 base pairs, but it would be a big thick band because it would be thicker than than the other one. It'd be twice as intense. We'd have twice as much DNA there. And then for the last example, if a 500 base pair of DNA between the two restriction ends were deleted, how would the banding pattern in the gel differ? And so in this case, what we would have is we have the 2,000 base pair piece, we'd have 4,000 minus 500, and then we'd have 5,000. So 4,000 minus 500 would be 3,500 base pairs, and then 5,000 base pairs. And so what we'd have is we'd have the 2,000. We would have the 2,000 base pair piece. We would have a piece at 3,500, and we'd have that piece at 5,000. Okay, so that's how the gel electrophoresis work and how restriction enzymes work in general. So now let's move on to this worksheet. So before we get into this worksheet, because there's not a lot to actually solve on this worksheet, I want to kind of illustrate what happens when we clone or put in a piece of DNA into a plasmid. So a plasmid is a small circular piece of DNA that looks a lot like this. This is what a plasmid would look like. So all plasmids have a few necessary components. Where the foreign piece of DNA is going to be, that's what we call the multiple cloning site, also known as polylinker, sometimes called the MCS for multiple cloning site. So this one location has a whole bunch of different restriction enzyme sites, so we can pick and choose whichever one's most appropriate for our particular experiment. We have what's called an origin of replication. So this is when we get this plasmid into a bacteria. When that bacteria replicates, it will also copy the plasmid and pass that plasmid on to each daughter cell. So we'll be able to have every bacteria will end up having this plasmid within it. This AMP-R stands for ampicillin resistance, and so that ampicillin resistance is the ampicillin resistance is for it's what's called a selectable marker. So what we want to do is we want to select for only those bacteria that have a plasmid, and so we're in order to do that we can add ampicillin to our culture, and then whatever bacteria don't have a plasmid will end up dying, and then only bacteria that have a plasmid will remain. Now, the last piece that we have is what's called LAC-Z. So what LAC-Z is this is what's called a screenable marker. And so the screenable marker is when we cut our plasmid and we cut it and we say cut with EcoR1, if this is my plasmid and I cut it with EcoR1, that plasmid could just close back up again. So what we're going to do is we're going to take this plasmid, we're going to cut it with EcoR1, and then we're going to have, say, a sequence of DNA that also has some EcoR1 restriction enzyme sites. Okay. And so, and now, so this would be so, and then this could just be a bigger piece of DNA, and this is what, and this is just more DNA at this end, more DNA at that end, and then say I'm trying to isolate this 500 base pairs that's found in between these two EcoR1 sites. Okay. Now, we're also going to cut, just like we did before, right between the G and the A there. And so when I cut this piece of DNA, I'm going to end up with a fragment in my tube that looks like this. Now, if you'll notice, the AATT here is complementary to this TTAA, and this TTAA is complementary to this AATT. So the goal is to get this fragment right into there and get that piece of DNA into our plasmid. So we're going to cut our plasmid with EcoR1. We're going to cut our DNA with EcoR1. And then what we want to do is we'll mix the two together, and then hopefully some of these pieces will end up into this spot with those compatible sticky ends. And we're going to add the enzyme ligase, which will seal up the phosphodiester backbone. So it'll seal up the DNA, so basically glues it all together. All right. Now, the trick here is the fact that this circular piece of DNA, this may be 2,000 or 3,000 base pairs in size. It's like having a rubber band. Okay. Now, and with that, I have sticky ends that like to stick back together again. Okay. And so the likelihood of this AATT finding this TTAA here is really good. Okay. You're going to very often just have that plasmid close back up on itself, and when we add ligase, it just gets sealed back up, and we have circular plasmids that have no inserted DNA. They're not of much interest to us because what we're trying to do is get this piece of DNA into the middle of it, so we end up with, say, our plasmid with our 500 base pair piece of DNA stuck in the middle, okay, rather than just a circular plasmid with no foreign DNA. And so that's what that last thing in the in that last piece of the plasmid is, that LAC-Z. The LAC-Z is what's called a screenable marker. So what this allows us to do is to screen for what bacteria have a plasmid and an inserted piece of DNA, because we know every plasmid that's in our culture is going, or every bacteria that's in our culture has a plasmid, because we've added ampicillin. That ampicillin is going to kill off anything else that is left in the tube that doesn't have a plasmid. So what we want to be able to do is differentiate between those plasmids that just close back up on themselves and those that end up with an inserted piece of DNA. So LAC-Z is the enzyme beta-galactosidase. This is what E. coli use to break down lactose. Now, what we're going to use this for is this chemical called XGAL is what's called a lactose analog. So a lactose analog means that the enzyme beta means that it has a molecular shape similar to lactose, so similar that beta-galactosidase is able to grab it and break it down. When beta-galactosidase breaks down XGAL, though, rather than breaking it into sugar components, when it breaks down XGAL, it causes a blue precipitate. Okay, so what this does is when we take our bacteria, we're going to cut up our plasmid with EcoR1. We're going to cut up our DNA with EcoR1. We're going to mix the two together. We're going to add ligase. That's going to glue everything together. So now we're going to have a tube filled with a bunch of plasmids. Some of the plasmids have foreign pieces of DNA stuck in our multiple cloning site. Some of them don't. We're going to take some of those plasmids and we're going to transform, which is the process of getting that plasmid into the bacteria. We're going to transform our bacteria so the bacteria uptake these plasmids, and then the bacteria will have these plasmids. We're going to add ampicillin. That's going to kill everything off. Now I'm going to have a bunch of bacteria with some with plasmids that have no inserted DNA, some with plasmids that have inserted DNA. I'm going to take some of that culture, maybe 20 microliters or something, and put it on an agar plate that has media for bacteria to grow on. That plate, agar is just it's a matrix, but we're gonna have food in there, and it's just like a solid thing that the bacteria can grow on that has food, and we're gonna just spread around those cells onto that plate. Okay. On that plate, we're also going to put ampicillin, and we're also going to put the chemical XGAL. Okay. So now we're going to have all of these individual colonies landing around on this plate. Now, the ampicillin means that if we have individual cells of E. coli landing around on this plate. If the E. coli doesn't have a plasmid, it's going to die. If it does have plasmid, it will live. On that plate is going to also be XGAL. And so now as that single cell divides into a colony of cells that are all identical to each other, if the colony of cells, they're all identical, and if they all have a plasmid with no inserted DNA in it, they're going to be expressing and producing beta-galactosidase that is fully functional. That fully functional beta-galactosidase is going to break down the XGAL, which will cause a blue precipitate, and so that colony on the plate is going to turn blue. Whereas if I've inserted my piece of DNA into the middle of it, and I have a piece of DNA in the middle of that beta-galactosidase gene, well, now beta-galactosidase, it might be being expressed, but now you have this giant piece of DNA inserted in the middle of it. It's not going to function properly. It can't process XGAL, so you don't get blue precipitate. So all of the bacteria that have no inserted DNA, all of those are going to be white. Okay, so this is why it's called a screenable marker. We're able to identify those colonies that have both a plasmid and an inserted piece of DNA, so that when we're moving forward in our experiment, we can pick those. Okay, so let's look at this worksheet and see what we got. Okay, so you identify population of frogs that have stunted growth and fleece-like texture to the skin. So then you isolate the gene responsible for this, called robin. And then, so you PCR amplify it and you sequence it and you identify that it's got two EcoR1 sites that are 1.3 kb away from each other. So what that means is like what I drew up here, where you have two EcoR1 sites that are 1.3 kb away. Instead of 500 base pairs, they're 1.3 kb, or 1.3 base, 1,300 base pairs away from each other. And then that has the open region. This is a purely hypothetical example, obviously. So you want to sequence and identify this. So you're going to cut your DNA, your PCR product that you have, that has these two EcoR1 sites sitting right on either side of your gene of interest. You're going to cut them with EcoR1. You're going to cut your plasmid, this puck 192 plasmid. You're going to cut that. Now the plasmid's 2.7 kb long. Okay, and then you transform your plasmid and plate them like I just described. Now draw the plasmid with the segment of DNA in it, and be certain the label where the EcoR1 sites would be. So this is a pretty straightforward. This is really what we're going to do is we're going to have 2.7 kb, and then we're going to have 1.3 kb. This is presumed to be Robin gene. That would be an EcoR1 site, and that would be an EcoR1 site. Okay. Now, the next question is what we call an insert check. So after you've done this process, you got to make sure that you actually have a piece of DNA here. And so what that means is you need to treat this, grow up some of this bacteria, extract out the plasmid, treat that plasmid with EcoR1. If you treat it with EcoR1, you're going to cut that 1.3 kb out. If you do that, and if you actually have a plasmid that has a 1.3 kb insert, you're going to cut at this site and at that site, and you're going to have two pieces of DNA, this 2.7 kb and this 1.3 kb. Now, a lot of people are probably going, how'd you get two EcoR1 sites there? Okay, because that's a common question. A lot of people, it takes a minute to recognize where those came from. And so why I have two EcoR1 sites on either side, where I started with one EcoR1 site here, but I now have two on either side. So we look at the diagram that I drew up here. My DNA got a little bit separated here. Let's move this. So if I cut this DNA, and now I have this AATTG is compatible with here, and this TTAA is compatible with there. When I insert this into my plasmid, I'm going to have AATTT, and then I'll actually have a CG here. And then this will be TTAA. And then right in there would be our EcoR1. And so if I drew this a little bit bigger, it would have been a little bit better. But so if I G C T T A A, and then we'd have C G T T A A, and then so it'd be A A T T, and then C G, and T T A A, with G C, and then right here that'd be 1.3 kb. This is an EcoR1 site, and this is an EcoR1 site. Okay, and so now what you'd do is you'd have all of these colonies on a plate, a bacterial plate. You'd grow them up. You'd pick a bunch of colonies, grow each one in a different tube of media, extract out the plasmid, and then you'd check to see which ones have an inserted piece of DNA. So you're going to treat your extracted plasmids with EcoR1 again, and you should cut them. Now, all of those cultures, and then you run whatever you have left on a gel, and visualize the DNA, see what you have. So if we ran this out on the gel, and this is what we had and what we were hoping to get, what we should have is a band of DNA that's 2.7 kb in size. And we should also have a band of DNA that's 1.3 kb in size. All of the white colonies on our plate should have two bands in them, okay? They don't always have to be the 1.3 kb, because we may cut up, there may be multiple, if we were, say, cutting up genomic DNA, we could be cutting up all different sizes of DNA, okay? And so we may have all different pieces of different size fragments, and each plasmid may have a different piece of DNA in it, and we may be just looking for the 1.3 kb one. Okay, but any white colony should have two bands when we do an insert check, because we're going to cut out the inserted piece of DNA. We're going to have one band that's the size of our plasmid and one band that's the size of the insert. Every blue colony should only have one band in the gel, and that one band should be 2.7 kb. Okay, because that's so they all should share that 2.7 kb in this example, because they all have a plasmid. We know that. We added ampicillin. So the only thing that can survive in terms of bacteria are the ones with plasmids. So they all have a plasmid. Now what we're looking for is the ones with two bands, and if those two bands is the second band the right size. Okay, so this is the last question to this is what would be next. So here you're checking, and you pick a colony from the plate, extract the plasmid, digest it with your DNA interest with EcoR1, you run it on the gel, and you get this. So what would you have hoped to see? Well, what we would hope to have seen is we would have hoped to see, so this two right here, this would be the plasmid. Okay, what we would have hoped to have seen would have been another band down here, because that would have been where our inserted DNA is. But since we only have one band that's 2.7 kb, it turns out that colony, maybe we mistakenly picked a blue colony. This should be a blue colony if is only the one band on the gel. Okay, and what color colony should you choose the next time? You should pick a white colony. Okay, so that's plasmid cloning in a nutshell, with restriction enzymes and how I use it. There are lots of different applications of plasmid cloning, lots of different variations and nuances of ways that you can do this in different contexts, like expression vectors and stuff like that. Most of the time, what we just described here, what its application is a way of isolating out a piece of DNA and growing up a whole lot of that piece of DNA, and so that you can do other stuff with it, whether it be sequencing or using it in other experiments downstream. But it's a way of getting a lot of the material that we need. In another video, I'll go over another technique that we have for getting a lot of material to do experiments with, and that's PCR, polymerase chain reaction. So there you go, folks. Restriction enzymes, plasmid cloning, all in a nutshell. Have a great day.